Models are the building blocks of science. In fact, it is the building block of science and the scientific method. We use the scientific method as a model to compartmentalize the steps and understand what we are supposed to do as we are moving from asking questions to posing hypotheses, designing an investigations procedure and apparatus, collecting data, processing it, interpreting the results, and evaluating all aspects to communicate the results and refine and/or revise the investigation for further research. Basically, a model is a substitution or a simplification of a system or a stand-in for a prediction. The key part to a model is it has to be similar to what it represents. For example, a model could be a cake recipe. It represents the process of making a cake. A model can be a chapter title, representing the chapter contents. It could be a roadmap of Madison, Wisconsin, representing the paths of transit in the city itself. The models can be used to make predictions of what will happen when the model is used. In the roadmap example, it can give you an idea of how long it will take to get from the state capitol building to Camp Randall Stadium to see the Wisconsin Badger football team win against the Minnesota Gophers. Moving towards science, models are just as common and do help us to make predictions about what will happen. They're really common and useful. However, the only thing that will allow models to work appropriately is that evidence must be collected to continue supporting a model's effectiveness. As models are used to represent the real world, the models we use are products of their time. As such, there are alternative models for the same thing. Our focus for today's video is on membrane structure. Since the 1920s, at least 3 models have existed to predict and understand how the plasma membrane functions to not only provide protection, but also restrict the passage of materials in and out of the cell. During the 19th century, experimentation suggested that the plasma membrane was lipid in nature, but did have a polar component as well. To save for time, we aren't going to get into that, so just take my word for it. Everett Gorter and Francois Grendel in the mid-1920s hypothesized that the plasma membrane must be a bilayer. They based this on calculations that the surface area of the lipid structures would be equal to twice the surface area of the cell membrane of an erythrocyte or red blood cell. Using red blood cells from a variety of mammal sources, Gorter and Grendel carefully spread the lipids into a single layer on the surface. Their data supported their hypothesis that the plasma membrane is a bilayer. However, these models are nonpolar hydrophobic compounds that form discrete structures when in the presence of water. They used these calculations in previous research by Dr. Irving Langmuir, and Gorter and Grendel developed the current model of the phospholipid bilayer. As far as membrane proteins, Gorter and Grendel identified they consisted as part of the membrane, but were unsure of where they fit into the model, resulting them not including it in their final product. Shortly after, in 1935, Hugh Davson and James Danielli extended the research of Gorter and Grendel to include protein in the plasma membrane model. Part of the reason for this is the bilayer is so incredibly thin yet protective. Due to the limitations of then present-day tools and apparatuses, they carefully limited their conclusions to experimental evidence. The evidence showed that most proteins are water-soluble and often attached to the surfaces of the membrane. These proteins were also amphipathic and interacted predictably with polar and nonpolar substances appropriately. The resulting Davson Danielli model suggested the bilayer was lined on either side by a single layer of proteins. By the 1950s, microscope technology advanced to the development of electron microscopes, which provided much higher resolution than light microscopes. David Robertson recorded electron micrographs, which led to him providing support for Davson Danielli's sandwich model of one layer of protein on either side of the lipid bilayer. Then, in the early 1970s, Jonathan Singer and Garth Nicholson, through careful experimentation and observation, were able to better explain the presence, structure, and positioning of proteins in the bilayer. There were peripheral proteins only interacting with the polar surfaces of the bilayer. There were integral proteins that went part or all the way through the bilayer, making up an irremovable component of the membrane. And lastly, all parts possess the capability to freely move through and around the bilayer. This was due to multiple experiments that produced strong evidence, which resulted in falsifying the Davson Danielli model. Whenever a model or theory is falsified, it needs to be replaced with a better one. In this case, we received the singer-nicolson model. Since, there has been further research through the advancement of technologies. While the singer-nicolson model, aka the fluid mosaic model, is generally widely accepted and still used today, it will be continually refined and developed into different forms, which will allow us to predict the structure and function of the plasma membrane.